O sea, la que nos está llevando el vídeo nos diga que adelante.
Hello, good afternoon, muy buenos días a todos. Es un placer estar hoy aquí de nuevo en el Instituto Cervantes de Sydney. It's an honor to be here in Instituto Cervantes in Sydney. We will continue in English. This is once again a collaboration that we put in place with SRAP, our fellow collaborators and friends from the Spanish Researchers Association in Australia and Pacific. It is once again our honor to host an activity aimed at for the promotion of Spanish scientists here in Instituto Cervantes de Sydney during the Science Week here in Australia. Once again, SRAP and Instituto Cervantes of Sydney put in place collaborations with our fellow scientists. In this case, it is an honor to have here with us the president of SRAP, Dr. Ángel López Sánchez, as well as Dr. Pablo Corcho-Caballero, here again, once again, Dr. Marcos Roca-Cusachs, and Dr. Maria Jones-Caballero. All of these colleagues will put in place several conferences aimed at the research promotion of Spanish researchers here in Australia. And it is, as I said before, an honor to have them here in Instituto Cervantes de Sydney in the framework of La Cátedra Cervantes, put in place here in Australia for the promotion of the researchers, of the work of the Spanish researchers here in Australia and the Pacific. Without further ado, I would like to pass the floor to the president of SRAP, Dr. Ángel López Sánchez. Thank you so much.
Thank you very much, Coral, for that introduction, and thank you very much, everyone, for coming here today. Can I have my presentation ready? As Coral have been mentioning, I'm the current president of the Spanish Researchers in Australia Pacific Association. And I need also this for passing the presentation. And SRAP, it is an association of around 250 researchers working in many different kind of research areas. See if I can see that. And not only here in Australia, but also in New Zealand. We have members in New Zealand. We have members in some of the Pacific Islands, in Vanuatu, Fiji, and Samoa. And we have several delegations, particularly here in Australia. We have New South Wales, Victoria, Queensland, and we have ACT, Western Australia, and hopefully we have the presentation running in some moment. There it is. There we go. Thank you very much. Here we are. No, well, did it twice. And as I was mentioning, SRAP particularly, I would like to define it as an independent and not-for-profit organization of volunteers in Australia Pacific. And SRAP was founded by people who are doing research in all branches of science, culture, and art. Do you mind removing the thing? Thank you. As I was mentioning, we have several delegations. I didn't click there, but anyway, okay. Let me tell you a little bit more about the mission and the objectives that we have. Our mission is fostering the collaboration and culture through research and development. And we have four main objectives that we have redefined during 2023. The first one, it is promoting and mentoring our researchers. The second one, it is scientific and cultural dissemination. For example, the activity that we are having here today in collaboration with the Instituto Cervantes. The third objective will be connecting academia with industry and society. And finally, promoting international relations and science diplomacy. We actually have plenty of activities, but one of the things that we like to do most, it is actually promoting the research and the different kind of work that our members have been producing. For that, we have our webpage with regular posts in the blog with several of this kind of research areas that different people have been doing at SRAP. And we also have launched the second SRAP Fundación Ramón Areces Early Career Research Awards, that is for 2023 for young researchers, just for promoting them and giving a bit more extra visibility. But as I was saying, we have plenty of activities that we realized, we performed during all the year. Not only what we call the research bites, that are internal talks among us, just for explaining what are we doing, the kind of research that we are doing. Also events connecting with the science diplomacy, for example, the Smart Spina Award that last year we had in the Academy of Science, Australian Academy of Science in Canberra. We have online events, and this one, it is a hybrid event, for example, today, that was several years ago. We also do organize visits to research facilities. For example, this is the visit that we did in 2022, that was last year, or 2 years ago, 21. In 2021, to Side and Spring Observatory, that we actually stay there during the night and see how the astronomers were actually observing. And we also organize other kind of big events, for example, this one, but for the Pinchos of Science, and connecting academia and industry, that we also perform some of these activities in collaboration with the Instituto Cervantes. In particular, I would like to emphasize that today we are having our first, the first of the 2 events during Australian National Science Week. On Thursday, we are having these Pinchos of Science, Brains and Bites, navigating the artificial intelligence revolution in education in the Village Choco with several of our collaborators, including not only Instituto Cervantes of Sydney, but Ita Spanish and Nikis Paelles and the Village Choco and the Spanish Embassy. And I will encourage you to attend to this event because it's going to be a lot of fun. Plus, you have Pinchos, and we are going to have a very easy atmosphere talking with key experts about how it is the situation in something so crucial right now, like in artificial intelligence. And a lot of ChatGPT will be in there. In general, SRAP have a very broad landscape with plenty of connections with different associations and research centers, not only universities in Australia, but also international research with a collaboration with Euraccess or RAICEX. RAICEX, it is the network of Association of Spanish Researchers working abroad. And 20 of these associations, SRAP, it is one of the founder members, but with culture and education, not only with Instituto Cervantes, but also with ALCE, the Linguistic and Culture Spanish in particular in Australia, and the Spanish Diplomatic Admissions, Australian Academy of Science, and not-for-profit organizations, for example, Casa Asia, the Fundación Española para la Ciencia y Tecnología, Fundación Ramón Areces, and a strong connection with industry. With Navantia, TIPSA, Iberdrola, Acciona, Indra, Ferrovial, our main collaborators, and particularly with La Cámara, the Spanish Australian Commerce Chamber. Finally, let me give you the opening, at least, of our big event that we will have the next 3rd of November. That is the 9th Australian Spain Research Forum, A Connected World. That will be at Macquarie University and also online. And in this forum this year, that is the 9th of the series, that will be mainly focused on the triple connection between academic research, pure research, with the research that industries are doing, and also the importance of science diplomacy. So the 3 aspects are going to be very much connected, and we have a very interesting program full of panel discussions and very key talks about different topics, and I will recommend you to go and check the details and just try to come for the day, because it is actually free. It's part of all that. And with that, finish my very short presentation just about the situation of SRAP, and I will give back the microphone to Coral for her to start presenting and introducing our speakers. Thank you very much for coming.
Thank you, Angel. We will proceed to continue with our program. So first of all, we have the honor to have here with us Dr. Pablo Corcho-Caballero. Pablo has been here before, and we have, yes, but it is an honor to have you as one of our key speakers to celebrate the Science Week here in Australia. So Pablo Corcho-Caballero was introduced to astronomy when he was at a teenage, in his teenage years, and after finishing his degree in physical sciences, he received his doctorate from the University of Universitat Autónoma de Madrid, Autonomous University of Madrid, and also the Macquarie University here in Sydney. His research focuses on the study and the formation and evolution of galaxies, and he is currently part of several international campaigns dedicated to the observation of galaxies through the use of new telescopes and instrumental techniques. And I am afraid to say he will shortly not be with us here in Australia, but it is an honor for you to be here with us today and to have shared the time that we shared together, and hopefully we will continue to collaborate here in SRAP with you in some other way. So the floor is yours, Pablo. Thank you so much once again for being here in Instituto Cervantes in Sydney.
Thank you very much for your words, Coral. I'm extremely happy to be here with you all. Hola, para los que me estén escuchando desde España, buenos días. I'm Pablo Corcho, and today I'm gonna present you some of the topics that I've done during my research, which is focused on understanding the evolution of galaxies in the universe. Just to introduce myself a little bit, I did my co-tutel PhD under a co-tutel agreement between the Universitat Autónoma de Madrid, as Coral said, and Macquarie University. Supervisors were Jaguas Casibar, which is now at ICRAR, but also I had the pleasure to have as my supervisor, Dr. Angel Lopez Sanchez, which is who is here with us. So let's get to it. The topic today is how do galaxies die? Because we think that life might be very difficult and harsh with us, but galaxies also struggle to evolve across the history of the universe. So I'm gonna try to show you just a glimpse of how galaxies, or the perils of the universe, and how galaxies struggle to form their stars. So let's get started by just having a brief introduction about the history of the universe. Well, if we think that the universe started with a Big Bang 14 million years ago, we have to wait 400 million years until we are able to see the first galaxies to form in the universe, until the dark matter is able to condense and give rise to these huge structures that will harbor life, that will harbor our place, the Earth, like now. So after waiting for these 400 million years, first galaxies will form and they will dominate the universe. They will become the atoms of the universe. And that's what we see nowadays. But the question is first, how or what are galaxies made of? What are the components of galaxies, right? I'm gonna go through the most trickiest components to the easiest ones, right? So the first one is dark matter. What is dark matter? We don't know. We have no idea, no clue. But dark matter is fundamental. Without dark matter, there are no galaxies. The universe could have not been able to evolve as it is nowadays, as we see it nowadays. Galaxies are thought to be embedded inside dark matter halos, and thanks to that, they are able to attract gravitationally all the material that they will process eventually. Also, galaxies contain a lot of dust. Yes, dust particles. Here you can see an image of a dust particle under a microscope, not a telescope, a microscope. The size of this image is 5 micron. Think like your hair width is like about 1 to 2 microns. So this is roughly 3 or 4 hairs of your hair, or of your head. But thanks to dust, galaxies are able to process all the material. And if you look at the night sky and you see the Milky Way, you will see a dark patch in the sky. That is nothing but the dust, absorbing all the light emitted by stars and producing these dark stripes in the sky. Thanks to the dust, gas is able to collapse and give rise, well, it's behind this banner, but give rise to beautiful structures like the Horsehead Nebula, where stars are born. Also, fundamental for galaxies is the accretion of gas. Galaxies harbor a lot of gas. Here you can see, for example, several galaxies from a survey, which is called THINGS, and these galaxies have been observed with interferometric radio observations that consist of a lot of antennas all put together to obtain these images where we can trace the emission of gas in these galaxies. And finally, you all might be thinking, what is the most important component of a galaxy? What is that we can see in the night sky? Obviously, stars. And I want you to take a moment and see in this picture, because you may think this picture, it doesn't have very good quality because you see a lot of noise in the picture. But the fact is that this is not noise. Those are stars. Every single point that you see in this image is a star. And this is the nucleus of a galaxy. This is just the nucleus of the second nearest galaxy to us, the Triangulum galaxy. And you can see the image on the right corresponds to this small, tiny red square on the left. So galaxies harbor hundreds of billions of stars, and they are the fundamental component of a galaxy. So all this research is trying to understand how do galaxies form and stop forming stars. Well, there is a life cycle, like everywhere, right? Like the rain that comes from the sky will precipitate and so on. Galaxies form their stars by the collapse of molecular gas clouds, and then they will form the stars, and then the stars will die, and they have this duty cycle. However, there is something that might go wrong. And what is this? Well, this is the topic of today's talk. The Killers of the Galaxy, Volume 1. Now I'm going to talk about the quenching of a galaxy. This is the term that we scientists use to refer to galaxies that die, that stop suddenly forming stars. When a galaxy quenches, it can be by two main factors, external factors or internal factors to the galaxy. This corresponds to external factors. The picture on the right that you see here, or on the left, sorry, comes from a numerical simulation. This is not real, this is not an image, this is from a computer, and this is extremely cool because we are able to simulate the universe on large scales. Now, with this, we can study the properties of environment around galaxies. When I say environment, it's because galaxies are not alone. In this picture, you don't see a single star, you see galaxies. This is a cluster of galaxies. This is the Coma cluster, a very nearby cluster of galaxies. These all galaxies have to live together, and life can be a little bit problematic when you have so many neighbors. On top of that, when those galaxies are all together, they are orbiting, and they are hot. They are creating a very hot environment. So this picture on the left corresponds to an image, to the optical image of the galaxy, of the cluster. But the picture on the right is an X-ray image of the cluster. This purplish emission that you see here all correspond to the gas that is inside of the cluster. And that gas, it's at millions of degrees. So it's extremely hot. And what happens when a galaxy falls into a cluster, into such a hot medium? Well, let me skip this. When a galaxy falls into a cluster, it will happen the same as when a meteor falls into the atmosphere. When a meteor falls into the atmosphere, you can see that it disintegrates, it gets destroyed because it impacts the Earth's atmosphere. Galaxies will experience the same in the cluster. And here you can see a beautiful jellyfish galaxy. That's the name we use to describe these beautiful systems. It's a galaxy that is falling towards the center of the cluster, and all the gas is stripped off. We call this process ram pressure stripping. And that's why these galaxies can get devoid of gas, and then they stop forming stars. Moreover, as I said, these galaxies live in very crowded regions. So when you are a little galaxy and you are located close to a very big, massive galaxy, that galaxy won't let you live easily and quietly during your life. That galaxy will harass you, will try to steal all your material because you are very small. And the galaxy will end up engulfing you, eating you because it's super big. That's what the Milky Way is currently doing with several dwarf galaxies. Now, for the second part, the internal processes, fire in the hole, because we all know, or scientists know, that inside of galaxies there is a very dangerous thing, which is a supermassive black hole. Supermassive black hole is a tiny thing compared to the whole extent of the galaxy, but it has the mass of that system is millions of times the mass of the sun, and it produces gravitational fields. It will trigger a lot of processes and will have a net impact, a huge impact on the evolution of the galaxy. So when you have a black hole in the middle of your galaxy and it's accreting material, it's acquiring all the material and it's creating a very hot halo around it, it will eject the material. Not only sinking all the material down, no, it will also eject the material outside, pushing outside the gas of the galaxy. And that's why you have these so-called jets of gas. This is the galaxy. This is very small compared to the size of these jets. That's all the material that is expelled from the center of the black hole. And eventually that will expel all the gas of the galaxy, but also will heat the gas. And that's very bad to form stars because we need the gas to cool, to settle down, to collapse. And if you are heating constantly the gas, it will be horrible. And finally, even star formation can kill star formation. Imagine if we have a galaxy that all of a sudden forms plenty of stars. We call that a starburst. After that, stars will eventually die. And when stars die, many of them, or some of them, will die as a supernovae. Those explosions will also generate these jets and will expel the material out of the galaxy. And sometimes the galaxy won't be able to acquire them again. So the galaxy again will get devoid of gas and eventually will die. So my conclusions for this talk are kind of connected to real life in a very silly way. So we know that the life is not easy, and well, galaxies even struggle in the universe. We know that galaxies have to co-live with thousands of systems in extremely crowded environments. The take-home message: be respectful with the people that surrounds you. When a galaxy is falling into a cluster, the gas might even get stripped. So take it easy, go slow, don't go that fast, enjoy the moment. Now, supermassive black holes, we have seen that can emit so much radiation that they are able to expel the material of the galaxy and hit the halo. It's fine to have problems, we all have, right? Finally, if the galaxies form too many stars at the same time, the supernova explosions can also produce the same effect. So we need to have a balanced diet and not eat too much, especially at night. With this, what I want to say is that, well, the take-home message for astronomy is that it teaches us many valuable life lessons because we always look on the bright side of life. Thank you very much.
Thank you so much. That definitely made the goal to, for us to understand it, even if we're not experts in the field. Second, we have here with us tonight Dr. Marcos Roca-Cusachs. By the way, if that's okay, we will always handle questions at the end, if that's okay. So as I said, I was saying, Dr. Marcos Roca-Cusachs, it is a privilege to have you here today with us. Mr. Dr. Roca-Cusachs is currently a postdoctoral fellow at the University of New South Wales here in Sydney. He completed his bachelor's degree in biology and master's degree in biodiversity University at the University of Barcelona and obtained his PhD in South Korea. He has ongoing collaborations with more than 8 international academic institutions and has an extensive field experience, being also the organizer and leader of 6 research expeditions and a team member of several more. He will be here today to talk about Pokemon, Gonna Catch Them All. Thank you so much.
Good afternoon and buenos días a todos desde España. Thank you very much for giving me the space to try to explain a bit what I do in a less scientific way, which is I'm not very used to that, but still. I'm sorry for that. So yeah, I'm gonna give a bit of a chat about what I do as an evolutionary biologist or evolutionary taxonomist, as I wanna call myself, and putting it a bit on real life, on things we might have come across in our life. So yeah, this is me. I'm Marco Roca-Cusachs. A bit of my career, it was said before, but graduated in biology and did a master's in biodiversity in Barcelona. Did my PhD in South Korea in Chungnam National University. Been a couple of years as a postdoc in Barcelona. And since this March this year, so very recently, I just moved here to Australia to do a postdoc research at University of New South Wales to work on the BMSB, which is a species that you will see further on in the presentation, which is an invasive species with a big interest, economically speaking, worldwide. So, as you have seen there before, I'm evolutionary taxonomist, and most of you evolutionary, you might have heard before, but most of you have probably never heard the word taxonomy. Taxonomy is basically the science that names, describes, and classifies organisms, so the whole pack of biodiversity, from big plants to the tiniest animals of them all. Yeah, and then still, if we're studying biodiversity, what are these main things that biodiversity study, or the units that we measure within biodiversity? These are the species. In this case, I wanted to give a whole spectrum and something that we might understand easier in our daily life as Pokemon. They're species that we have come across if we have kids or so along oral life, and my generation as youngers, we've all played with them. So how do species evolve? So the diversity we see nowadays has come through many processes, and all these processes through diversification, natural selection, and extinction have led to the current amount of species we currently have. All individuals are naturally variable, and this natural variability that we find in the groups of species provides individuals with better traits that suit them better or less to different environments. The diversification events that may cause the species that we have now are mainly due to species that come all over from the same ancestor or either the same colonization event. Then some important thing we have within the creation of species is natural selection. We've seen this a lot with Darwin in the past. He did a lot of research regarding natural selection. And natural selection is a process through populations of organisms adapt and change, and therefore result in the different organisms that we have nowadays. However, if the environment is not suitable for these organisms, and therefore it creates a harsh environment for them, we can have events of extinction. And we now know that we have many species that have become extinct because we can go back to the fossil records. And through studying these fossil records, we know that we can more or less study the evolution that these animals, plants, other organisms were there. And additionally, through another part of the evolution of the world, which is through tectonics, which we can kind of understand how the patterns of the different continents worked, we can get these fossils and put them in a geological scale, and therefore understand where they fit in terms of the tectonical plates and in terms of the evolution, giving us a better understanding of the distribution of the current species in the space and time. Unluckily, nowadays, we are facing one of the biggest threats in biodiversity, and this is called the biodiversity decline. It is mainly due to global climate change, which we are not helping it by means of habitat loss, unsustainable resource use, invasive species, or pollution. Therefore, is where an evolutionary taxonomist comes in handy. We study taxonomy. We study what do we have there in the wild, biodiversity. So that's I listed here some cool parts we do, which is we go on amazing field trips, find new species, describe and name these new species. Then we go to our very old collections, our very old museums, check for the specimens, and dig in the material over there. And then we get to do some fun part, which is hypothesize about their phylogenetic relationships. You've been hearing, though, that I name species all the time, and it might seem very clear what a species is for what I've been saying. But in most of the cases, it's not even easy for us. As you might see here, the image on the right, there's several things that might look for you as different species. While on the image on the left, there's a lot of things that might seem exactly the same. In the first case, they're all the same species. They're just variable, and they look extremely different, though they act exactly the same. While the image on the right, on the other side, sorry, the butterflies, all of them are different species. And therefore, even not for us as taxonomy, sometimes it's not easy for us to determine which one it is which. And therefore, we need to do much more. So all the fun part from the slide before, it might not get that fun. So my research group is the family Pentatomides, stink bugs, chinches in Spanish, which is an economically important group. The reason why I got to this group, at this point, I really don't know. I kind of like them. They're stinky, they're big. Also had a good professor back home in Barcelona that she helped me a lot to get into this group. But also while I've been studying it, I've realized, and we know at this stage, that they are important because some of the species constitute important plant pests. Some are used as biological control agents. So in some cases, they are used to control other pests and therefore are good for crop control, etc. And in some places, we don't do back home, but South America, they do a lot. And in Southeast Asia, some of the species are consumed as food, giving us a lot of protein value to the diet in diets that are protein poor. And as you have seen in the slide before, which I'll go, plant pests, and this is the brown marmorated stink bug, which is the reason why I'm here in Australia. This is one of the biggest plant pest worldwide. It is officially not in Australia yet, so if you get to see it, contact us at any point. It's been found in the borders, but yet still not here in Australia. And it basically feeds on most of plants, most of vegetables, most of fruits, making these fruits and vegetables not consumable or not soldable in the market. Yeah, and then for all the for that I have been saying before, it's like part of our job, besides doing all that cool stuff I've listed, it's like we need to really concentrate and focus, and there's a lot of background behind. So part of it, it's the classical taxonomic approach, which is the one you might have seen more. It's we are under a microscope checking specimens, checking collections and doing dissections, and then describing the species and listing them for different inventories and stuff. Then we got more integrative approaches, which in this case include molecules, DNA sequences, also a lot of different morphometrics, geometrics, and different data, biological or geographical, in order to determine which one of its groups corresponds to a species, and even though make all the differences among groups we need to do afterwards in order to be able to catalog them properly. And then last but not least, and maybe this one might sound a bit more ethereal for you, or maybe not, is we do phylogenies of these species. A phylogeny is basically reconstructing the family inheritance of these species and going backwards in time and finding them, finding their closest relatives. It would be like a kind of genealogy of these bugs in a worldwide context. And this was part of my my PhD, and it's also part of my whole life career. And even though that was my whole PhD worth, we did not find the relationships among them because the data we were using was still not enough. So after that, during my postdoc in Barcelona this last year, we combined a whole bunch of other data sets, not only produced by me, but also produced by several other colleagues from all around the globe. We decided to get all this information together, put in a big data set, run it again with, as you see, a bunch of different species, more than 200 species. And this was presented in the meeting of the International Dermatology Society last summer. And as you can see, there was not much conclusion up to it. And again, we still have no idea about what's going on. And luckily for me, working with the brown marmorated stingbug, which is a group of insects that fits inside this group, has allowed me the possibility to come over here, discover where this should be placed in the whole tree of life of the pentatomy of the stinkbugs, and get to continue with my research for some, at least some years more. And thank you very much.
Thank you so much. Thank you so much for being here and for making such complicated areas accessible for those of us who are not scientists. We definitely do understand what Pokemon is, and definitely now can have a certain idea of the relation of your studies. Without further ado, last but not least, we have not only explored astronomy, explored biology, but now it is a wonderful opportunity to explore the area of dermatology. We have here with us a very active member of SRAP, as well as our previous members, Dr. Maria Jones-Caballero, dermatologist. She also graduated from the University, la Universidad Autónoma de Madrid, as well as Pablo and as well as myself in my case. She's a specialist in dermatology. She achieved her specialist in dermatology in La Princesa, a university hospital also in Madrid. She has been practicing since 1992. Maria runs 2 specialist clinics at Westmead Hospital, one for transplant patients and one for high-risk melanoma and oncodermatology patients. As I said before, it is an honor to have you here with us. And without further ado, it's your turn. Thank you.
Close to everyone can hear me? Very good. Okay, I have to advise to get started with. Well, first thing, I acknowledge the Gadigal of the Eora Nation, the traditional custodians of this land, and pay my respects to the elder, both past and present. And the first thing I need to say, if you feel uncomfortable or nauseous with images that can be a little bit gory or bloody, or just close the eyes and listen, only listen. Don't look at this. I mean, I was not sure if I really wanted to bring this, but I really want the people to know how bad a cancer can be. And then transplant patients offer us the opportunity to, unfortunately, to see that. So let's get started. Is this just... I have to point there. Okay. So this is well. We are talking about what happens to the skin when you get a solid organ transplant. Okay, and what does it mean that? That means that sometimes our organs. It can be any organ, but the most frequent one that we are doing transplants are the kidneys, but it can be the heart, the lungs, liver. So they can fail, they can not work anymore. And someone had a great idea saying, oh, this guy is not living anymore. Let's put the organ of the person that died in this person that wants to live. And that's what we are doing. And I think the idea is very impressive. But what happens that when you put an organ, for example, a kidney, on a patient, this organ does it have this? Is this? Do you have laser? No. The top is laser. Oh, okay. Okay. So this, let's say, this is the one of the cells of this kidney, and then it gets in contact with our immune system. Our immune system says, oh, you are not supposed to be here. Because interestingly, when we are born, we develop this immune system that allows us to recognize ourselves. But if someone from outside wants to come into us, says, no, you are not welcome. And this is usually everyone knows about the virus, the bacteria. Our immune system reacts against it. But it can react against other organs. And then we create what we call a graft rejection. We don't want that organ inside. It can be in an acute way, or sometimes it takes a little bit of time for the immune system to realize that there is an organ coming, and it takes a little bit time, but eventually you may create a chronic graft rejection. So what can we do to avoid this? So, okay, let's do the immune system lower. Let's make it work less hard, so they don't recognize that organ that is coming into our body as something that is weird. And then, can I be here? Because I thought I was going to have the, okay. So there we have the concept of immunosuppression. Immunosuppression means that we are reducing the activity or the efficacy of our immune system. And there are two main consequences that are going to happen when our immune system doesn't work well. In that case, because we on purpose are making the immune system not to work well, we are giving the patients a medication that reduces immunosuppression. One of them is infections. One of them is infections. Okay. This is infections, and the other is cancers, because cancers are all other things that we have inside and we don't like. When we develop, apparently, we are developing cancers in our body all the time, and our immune system is getting rid of them. That's the reason. If you are immunosuppressed, if you are depressed and the immune system goes down, then you develop more cancers. And then I'm going to divide the lecture in 5 parts. I'm going to talk a little bit about the cutaneous infections, the cutaneous tumors that happen in immunosuppressant patients. Then these medications can have side effects, and this is another problem that we are going to see in the skin because as a side effect of the immunosuppressant drugs. And then I will tell you a little bit how we treat these conditions. So cutaneous infections. Usually they can be bacterial, folliculitis, erysipelas, cellulitis, anything that happen on a normal person, but usually is more aggressive, more severe, develops quicker, not nice. Viral infections, it can be all the ones that we also have. We have warts, herpes, shingles, anything. Again, they have then much severe. So, for example, that's a cold sore. A normal cold sore is a little thing that you have here. Transplant patient will have the whole lip with a cold sore. The warts, you have a little wart here. They have, they have big warts that might cover big areas like these all warts. This condition is called molluscum contagiosum. Maybe if you have little kids, they get it in the pools. But they can be very a big problem because from the cosmetic point of view, you may have a lot of them, and it's very hard to live with all these things. Fungal infections, either a simple tinea, deeper infections, even that can be disseminated to the body, and they can kill you, actually. And for example, a normal ringworm that you just have a little bit, you are with a cat, with a dog, you have this little patch. Look how a tinea can be in a patient with a transplant. Look at, this is a patient of mine, like a really, really horrible tinnitus, very resistant to treatment. And then let's talk about cancer that is even a worse problem. So I'm going to talk about 3 main cancers that we are going to find, and this is good to know about, even if you are not immunosuppressed, you need to see, like, for example, a basal cell carcinoma is the most popular. It can be a nodule, it can be little thin, but it can be really little thin. And that's the role of a dermatologist, to detect very early, so we don't have to do big cuttings on those patients. Squamous cell carcinoma is really very frequent in these patients, and they can show as a cutaneous horn. Can we take the light here or not? Or because it's just focusing. I don't think that is well seen. No, okay, that doesn't matter. And this is a squamous cell carcinoma on the scalp, really big, as you can see. And this is a melanoma. You can see a variation of the color. And then, again, a summary of a typical basal, typical squamous, and a typical melanoma. But the problem is that this is a basal. Scar. Sometimes a basal is like nothing. Our role is just to detect and to be able to see lesions that they look like nothing. And this is a little squamous. This is a little squamous, a baby squamous. Be careful with them. And this, unbelievable, is a melanoma. Sometimes melanoma, they can be so nasty that they are what we call amelanotic, hypopigmented. They don't produce melanin. And then you have to have a high degree of suspicion and do a biopsy, so we'll see the melanocytic of the melanoma cells there. So, and another one that we don't see them very often, but is really increased the incidence, is Kaposi. We all know, we learned about this tumor when AIDS came to in the 80s. HIV also, it was another reason why the patients had immunosuppression, and Kaposi sarcoma is one of the cancers they can have. Interestingly, usually, basal cell carcinomas are more frequent in the population that is squamous, but in this case, it happens the opposite. It's more frequent squamous than basal in these patients. We don't know why. And the patients have up to 5 times more risk of having melanoma. The risk multiplied by 10 for basals, by 65 for squamous cell carcinoma, so it's a lot. You really see that these patients, some of them have a cancer every month, which is very appalling and very bad for them. So I just presented a couple of cases to see how quickly they grow in the skin. So keep on, yeah. So this is a case on the hand. And that was December 20. In March, look the nodules, and these are cancer cells that go to the lymph nodes and kill the patient. They look like a little wart, but they are really dangerous, and they can really kill the patient. So we have to be very careful. And when we see the patient in this stage, is when we have to act. We cannot wait for this. And this is a young lady, again, July, and 6 weeks later. The squamous eventually got lymph nodes with radiotherapy, and she almost died, but fortunately she's now alive still. Yeah, and then a few side effects of these medications. They are not related to the infections, they are not related to the tumors. Sometimes they can develop these things that we call sebaceous hyperplasias, mainly with cyclosporine. They look very much like molluscum, but they are different. You can differentiate it when you look at them with the dermoscope, our little magic telescope for the skin. Then sometimes you can develop acne, osteroids, hypertrichosis. Sometimes the hair grows a lot with these medications, or hyperplasia of the gums. It's really bad. And then let's talk a little bit about treatment. I'm going to go very quickly because Spain is plain, so I don't know what I'm doing here. Of course, we have to do a specific treatment, virus, bacteria, whatever, you do it, but do it quickly and specifically, either with a topical or with a oral or intravenous if needed. And the cancer, similar approach to what we do with any cancer. Let's cut it if we can. Sometimes we cannot, it's too big, or the area is very difficult to close. We can do radiotherapy. There are different topical treatments that we can use, and we can kill cancers with creams, which is amazing. And when it's metastatic or very advanced, we have to use new molecules that we call monoclonal antibodies. And we have to be careful because you can see this, nothing, it's a little basal. No, it's not a little basal. This was the amount of skin that needed to be removed because it was all hidden under. And there is this technique that we call Mohs surgery, that we cut a little bit, we check the borders. Oh, there is still here. I couldn't see it. We have to keep on cutting until the margins are clear. And the good thing about Mohs surgery is that you end up knowing and clearing the lesion because you map it. And sometimes the cancers can be really, really big. This was a metastatic squamous cell carcinoma. And the patients suffer a lot because the quality of life is very, very hard. And very, very important is that not only dermatologists are involved in this. We are in the first line. We have to diagnose them soon. We have to explain to them how to prevent that. But we have to involve radio-oncologists, oncosurgeons, we need to remove lymph nodes, and all the specialists, and I will explain to you why, depending on the organ that has been transplanted, they have their own specialists, and we need to be in close contact with them. And also radiologists and dermopathologists will help us to do better diagnosis. So very important, multidisciplinary group work. And then now prevention. So there is different ways. We can, for example, before a transplant, the transplant doctors make sure that they get all the vaccinations, so they are ready to fight any infection. Sometimes we have to give them antibiotics on a long-term basis, and also we have to explain to the patients that they are at risk of getting infections that other people wouldn't have. If they have an aquarium, they can have Mycobacterium marinum. So we have to explain to them they have to wear gloves, they have to protect themselves, be careful with the gardens. They have to not isolate it, not to be in a bubble, but be the hygiene has to be very good for them. And then, look, that's a very interesting article. It's a little bit old, but in Sydney, there was a study where skin cancer metastatic accounted for 27%, that's more than 1/4 of the post-cardiac transplantation death. So, of all the people after a transplant, in 4 years, 1/4 died because of skin cancer. So, and it's terrible to think the investment, emotional and in money that we put in those transplants to let the patients die because they had a little noodle that on time would have been cured. So that's our really role of dermatologists. We have to explain to the specialists, send the patients since the very beginning, even before the transplant. We are commencing to explain to them how they are going to protect themselves from the sun. We're going to explain, unless I'm going to tell you a little bit what we tell them. First, we need to classify the patient. Who is at risk? At risk will be like any patient that even not transplant, any person. You are you have more risk of cancer if you are old. If you are older. If your skin is fair with the blue eyes, you have lots of freckles. If you have had lots of sun exposure, especially if you have worked outdoors and you have a family history of cancer, or a personal history. These are the main risk factors that everyone has, and of course, a transplant patient will also have. But there are also risks that are related to they are unique for these transplant patients, and they will be the older the transplant, the higher risk. How many years you have been having this transplant? No, I had my transplant 2 years ago. I relax. If they say 30 years ago, say, okay, we're in trouble. Also, which age you have your transplant. If your transplant was done at a young age, usually not so much risk. And also the level of immunosuppression, depending on how much medication you are using and how low is your immune system, you will have more or less risk of cancer. And also having warts can be another risk factor. And then as a preventive measures, there is a medical management. We have vitamin B, vitamin A derivatives, and a few different anti-cancer creams that we put before the cancer develops to prevent. So we are to detect pre-cancer to avoid the cancer to develop. And then there is the second line is when all these things don't work and the patient still is having lots of cancer, then we have to talk to the specialist. That's the reason I told you we have to know the nephrologist, the gastroenterologist. I'm going to dinner this Friday to try to do a little bit of networking with those specialists, because sometimes we have to convince them to change the immunosuppression, put it lower, or change a different one that is not so favors so much the production of cancer. And then sun protection and early diagnosis are so important that I'm going to put a couple of slides for those, okay? So important, sun protection is the main strategy that we have to implant in the patient's brain, very important. And the problem is that around 40%, 50%, they don't remember even have been told ever that they have to do sun protection. And also, only 40% are using sunscreen. So this has to change, and this will change the panorama of the prognosis of these patients. Of course, everyone knows, I'm not going to waste time, cover yourself, it's not the sun protection, do it again, or, you know, all those things. I'm not going to go through them. Clothes are very important. There is even some laundry that you can use and add some sun protection on the clothes. And this is not enough. It's very funny. The favorite one in white t-shirt, a t-shirt white, no, this is not protecting you. You have to take whatever you want. If you want to know if you are protected, grab the cloth and see through. If you can see the sun coming, it's coming. Okay, so it has to be really opaque. And of course, avoid the outdoors activities and tanning boots, no way. And then, so that was the sun protection and also the early prevention. Early prevention is really, you don't prevent with that act, but you prevent that the cancer gets too big. That's what we pretend with that, intend with that. And so we have to explain the patients how to, most of the times if there is a family member, it's better to tell them how to organize very frequently, once a month, checking that is any new lesion coming. And usually they have to look for areas that are red, sandpaper. Skin cancer looks like dryness. People think they are having dryness, not that they are having pre-cancer. Sores that don't heal. That's the way we explain to them. Areas that are bleeding easily, or spots that are changing color, and the famous A, B, C, D, E. That is the guidelines for the moles that are changing into a melanoma. So the asymmetry of a lesion, you can see this, the borders being irregular, color also variations, the diameter more than 6 millimeters, but sometimes we find them smaller, and the lesion is changing. That's an alarm sign. And then I'm going to finish now just saying that the cutaneous lesions are very common in transplant patients, numerous, very complex, mainly, as I said, infections and tumors, very unusual and very aggressive, more than in normal population. and that a quick diagnosis and early intervention will give us a much better prognosis. And again, I will never be tired of saying sun protection is a must. There's a few references here. I think some of these ideas and slides come from this at-risk. What happened? It doesn't go to the end. Okay, yeah. Are you going? Can you do that? So this alliance of association of doctors, nurses and patients, and they work together to improve the quality of life of the patient. It's called at risk. And finally, I want to thank my 3 beautiful girls that are always supporting me, and I love them. and of you, I thank you for being here. You are really brave and generous.
Please sit down. No, con el micrófono, siéntate ahí. Sit down. Okay. Thank you so much, Maria. Thank you so much from my colleagues of SRAP. So we're going to around to an end. In this case, we would like to acknowledge the time of our fellow members of SRAP for being here today. As you can see, the areas of expertise are very different to try to reflect the variety and richness of all the members of SRAP. I would like to thank you for your time here today and to pass the floor for some questions, if that's okay. No.
Okay, I have to do a question. Do you have a question? Do you have a question? Go. You. Use the microphone.
Just a second.
Sting bags? Yeah. I have them in my garden. I don't think it's the same. The sting bags?
I mean, it's common you have stink bugs in Australia. I mean, you've got heaps of stink bugs. Sorry, now better. You have heaps of stink bugs which are native to Australia. You have some additional ones that should not be here, but at this stage we can't do anything. If you have the brown marmoratic stink bug, call us at university.
I'm just going to get rid of 3 citrus trees that I have, little dwarfs.
Okay.
Oranges. Trying to get rid of them at the early stage.
But they're
when
pretty,
they're green.
Pretty big ones, right?
Yeah, they're horrible.
They're local. They should be there.
Also, they're not.
I mean, they're not good for your tree, but they're not bad for the environment.
They demolish the tree.
How big is that one that you are looking for? How big it is? As big as that.
They're big.
Yeah, it's like 1.5 centimeters.
And by the end of summer, the trees, all the leaves are gone. So they're not very good.
They're not very good, but for what I say always to fellow colleagues, it's like they were here before us.
But then I can have oranges. That's the problem. Or lemons.
I mean, you can always like kill them. Soapy water would do. Or pesticide would do. Like I'm not a fan of using pesticides on native species.
Yes.
I do it with non-native species because they were not meant to be here. But I like my bugs, so I like when I go somewhere. I like seeing.
Not sting bags. They also smell terrible.
They do smell terrible.
And also, they seem to give you allergies. The smell when they linger on the air. Because I got an allergy now after trying to kill them for every summer. I tried to. When they were little, I just green. You know, they start green.
Mm-hmm.
I used this capsicum with soap, and they die with that. But somehow I managed to hide and grow into the big ones that look like monsters, like very thick, and they're hard to kill. You have to grab them by hand, and that is dangerous because they could spray the oil, which I think might not be good for you.
It's.
I have a patient. I was thinking about one of my patients came with the hands so yellow. So my patient came with the hands so yellow on one side, and I thought, it's like the smokers, but a lo bestia. Like, and a lot of yellow thing. And then I said, what are you doing with your hands? I just kill the things with the hands. So
No, you can. You have to use thongs.
He was not dead or anything. He had a horrible yellow hand.
I know.
And that was it, but he was okay.
I tried all type of things, and so now I have to
But
get rid of the
don't
trees.
try that. Ugh.
The trees will have to go, and that's sad. No more oranges. Well, I only have 2 or 3 at the end of the season because I eat quickly. As soon as the bud of the orange, the flower, then you have the bud. They go for it, and within one night, the next morning, everything is gone. They are terrible. So I just thought it was the same stain bags you're studying.
There's without a picture, it's difficult to say.
No,
I
it
think
was the first one you saw, the black ones. They look like.
I should see a picture.
They-
But
No,
we
I
can
don't.
keep this conversation later if you
Sure.
want. So
Well,
we
it
can.
doesn't matter.
But
I'm
if you
just
have
going to
if
get
you have a
rid of the
picture, I'll happily have a look at it and we can discuss it later.
I'm going to get rid of the trees. No trees.
Thank you very much for that interesting discussion. Let me ask a couple of extra questions to the other panelists. Going first with Pablo. Pablo, what is the strangest thing that you have seen in a galaxy?
Great question. What is the strangest thing? That's a very tricky one. Well, I mean, I haven't. The way I work with galaxies is not like you take a look at all of them, right? Because I work with thousands of galaxies, so it's impossible to visually check all of these galaxies. So we need to develop algorithms and techniques to check on that. So at the end of the day, what you see is just numbers and graphs, right? But it's well known that there are some weird galaxies that, for example, at the beginning of my talk, I said the fundamental ingredient for a galaxy is dark matter. But there are some galaxies that are found to lack dark matter. Why that galaxy don't have any dark matter whatsoever? And that's because some other galaxy probably stole it. Because the first thing that a galaxy will keep away from you is your dark matter, because it's the closest thing that the galaxy can touch first. And I think that's kind of the weirdest thing I can describe. Yeah.
Thank you. Maria, I have also a question for you. Don't worry. You have been talking a lot about different kind of cancers and situations in the skin, and mentioned some few of your patients. But I would like to know, do you have very different kind of people, different ages, or usually your patients are much more elders?
Because I work in Westmead Hospital adults, I don't see kids. So they are all over 16. And most of the patients, because the one that
But over 16 is still not kids, but you have-
Usually I am seeing some 18 getting ready for a lung transplant. Some on the 20 something, I see some of them, but most of the one that I see, my frequent flyers are 60s, 70s. Yeah, if they survive, I don't have them with 80 or 90. They don't survive to that age. So they are 50s, 60s, but with lots of cancers. Yeah.
Thank you very much. Do we have a very last question? Do you mind, Coral?
As a child, I was brought up on a farm and thought it was very de rigueur to chase sheep with just a pair of shorts on. So for years and years, I was running around getting nice and tanned, and I thought that was pretty good. It looked good on the beach. However, later on, I was told that you damage your skin at an early age, it remains damaged for the rest of your life. Or I'm asking you, does your skin repair and regenerate?
The skin regenerates, and there can be some damage in the background, but every single day that you sun expose, you add more damage to that DNA. So when my patient said, "Oh, this damage was done when I was young," and you keep on doing it, no. The idea is that you are born, imagine, with an empty jar, and then sun is like the water. You keep on putting water. At the beginning, you fill that jar, and nothing happens. But there's one moment that is really filled, and a drop spills the water. Another drop spills the water. Meaning, spilling the water, you get a cancer. So it's very important, even if you had a big amount of sun damage, even in that case, more important, because you don't have more room of water. Any little drop that you put will produce a cancer. So the patients that had a history of sun damage when they were young are the ones that have to be even more careful. They don't have that possibility, that room for a wait to see what is going to happen. You're going to get it easily. So patients have to protect all of them and more if you have had sun damage when you were young.
Thank you. Since I was on the farm, we've sold the farm, and I now put hats on.
Very good. I'm proud of you.
Thank you. Thank you so much. We have one of the last questions, if that's okay. With from an online viewer. The question is for Marcos, saying that the viewer is assuming that with new technologies, the way of identifying different species has changed with time. Perhaps these days you are not using that much of microscopes, but genetics tests.
We actually, I mean, I did, one of my slides showed that we currently use different genetic markers that are generally used to identify species, and we can identify most of them with precise genetic markers. In case of animals, we use CO1, which is a short fragment of around 600 base pairs, which is in theory unique for each animal. But as we might know in nature, this is never a black or white situation. You always have cases in where this marker does not show exactly what you want, or the evolution of this gene, which is CO1, is much slower, and therefore you cannot recover enough changes in the gene to be able to separate it when you do the analysis. There might be some species that, even though you, what we say, amplify this gene, you cannot recover any information because sometimes it doesn't work. You're basically there, you do it once and over again, and you can't get that information back. So you need to go always back to the classical part of checking these specimens. And also on the other side, there is some cases where they go to the Amazon rainforest, to Borneo, to the bush, get everything, sequence everything, and then they get 3,000 million sequences, but they cannot know what was what. They just have numbers. And then you need a specialist that basically can identify those samples, give them a code, match the code, and after that is done, you can code anything you want that is already been coded before and give it a name. But if it's not databased, it's impossible to track down because you will just be getting sequences out of it without even knowing what that sequence meant. So we still need taxonomies, contract us.
If we'll continue with the questions, this last question, if that's okay, because we're running out of time.
Yeah. I have a question about dermatology and the after effects of the immunosuppressant drugs. I would like to know if you can tell us that because of immunosuppressant drugs causing problems with skin and perhaps skin cancers, do they also cause cancers in other parts of the body?
Yes, the same thing. Yes, of course. If your immune system is not working, it produces cancer. They have lymphomas, they have more frequent breast cancer, bowel cancer.
Right.
They do produce all of that. The reason why it's so extreme and so frequent, much more frequent, the increase of cancer on the skin and in other organs is not clear yet, but it's much more, maybe the, I don't know, the sun exposure, I don't know, yeah, but.
The previous lesions and.
But they do have more cancers, they do.
Okay, thank you.
Thank you. Well, we are running out of time and coming to an end to our conference here in Sydney, in Instituto Cervantes of Sydney. Once again, we truly appreciate your time. As we mentioned before, this is a perfect representation of the variety and the richness of the members here of SRAP. Once again, we're here to promote, to celebrate the Science Week here in Australia. And once again, our work here today is done, but not at the least finished, because this is an ongoing process and effort that we are pushing through with our colleagues and friends from SRAP. This here today has finished, as I said, but we still have a lot to go, the pinchos that we will do with our friends in the SRAP and the Association of Eat Spanish, but much more. Once again, here this year, we will also be in the 9th Forum of SRAP, participating during the month of November, and Instituto Cervantes of Sydney will also help in any way that can be possible in the science diplomacy, and along with the Embassy of Spain here in Australia for the Premio de Malaspina. I would like to thank all our colleagues here for being here today, and if you would like to close, I will give you the last floor.
No,
here,
just I will just finish thanking you, the Instituto Cervantes Sydney, and for allowing us to be able to tell people, to tell the community, the society, all the very interesting kind of different research that our members are doing, and we really are looking forward to all these other collaborations. Thank you very much for that, and thank you all of you for coming here today, and the people who are listening to us online here in Australia, in Spain, and overseas. And yeah, thank you very much, everyone. Thank you.